Today we will be presenting Clarity through Clarifi Salivary Testing for Autism. My name is Erin and my other group members are Summer, Jamie, and Diana. Previously, the diagnosis of autism was based solely on behavior and characteristics of a child or adult. Reliable testing for autism has been unachievable until now. Clarifi is a diagnostic test that uses biomarkers in saliva as a way for early detection and diagnosis. Methods used in saliva testing will be further discussed, analyzed, and compared in our presentation today.
Hi, my name is Jamie. I'm here to talk about the problem with the diagnosis of autism. Most individuals might agree that they want the best for the next generation, especially new individuals in a growing family. Making sure that children are healthy, happy, and able to have their needs met is a substantial component when it comes to making decisions on education, literacy, development, and preparation for children. Unfortunately, some developmental obstacles are easier to recognize and diagnose than others. But what if there was a non-invasive way to check for abnormalities in children earlier, giving parents and caregivers access to care as early as possible? As science has evolved, so has an understanding of disorders in children and adults and the relationship of addressing issues earlier in life. According to the Center for Disease Control and Prevention, 1 in 59 children are diagnosed as being somewhere on the autism spectrum. The CDC also states that the ASD can be diagnosed as early as 2 years of age through observation and various tests. However, many children are not diagnosed until the age of 4. While some individuals argue that mental difficulty can be associated with the nature versus nurture viewpoint, having a scientific test could in fact show that individuals have a predisposition or genetic marker for ASD. Currently, autism is thought to have some type of genetic connection, but is not deeply understood.
The purpose of our research today is to present a new and reliable option for early detection and diagnosis of the autism spectrum disorder. Clarifi's non-invasive testing can be performed on a child as early as 18 months of age.
Hi, my name is Diana and I will be going over methods today. To find if Clarifi will be beneficial in diagnosing a child with an ASD diagnosis, various methods were used during the study of salivary testing. The components of the testing included study population, data collection, RNA collection, quantification, processing, data normalization, machine learning approach, functional studies, and finally a comparison of results. This cross-sectional study included 456 children, ages 19 to 83 months. Also, medical and demographic information was collected from the participants, such as sex, age, gestational age at birth, family history with ASD, sleep disorders, existing mental health disorders, gastrointestinal diagnosis, and asthma, followed by an examination with medical professionals. In the study, microbial and human RNA were measured in the saliva of all the participants. The whole saliva was gathered in place in a highly absorbent swab at the base of the tongue, near the sublingual ducts, and bilaterally between the gums and buccal mucosa, near the parotid ducts. Then the association between microbial RNA and human abundance were determined with Pearson correlation analysis and the clustering with complete linkages. These samples were then divided into random sets and their RNA, then divided into 5 subtypes of RNA. The subtypes were then compared to develop the RNA-based prosecutor that could distinguish the ASD children from the non-ASD children. With the salivary RNA abundance study, they have developed Clarifi, the non-invasive test that could be beneficial when diagnosing a child with an ASD diagnosis based upon their RNA biomarkers. Within the given study set, the data collected indicated that out of 456 children, 134 had neurotypical delays, 238 had an ASD diagnosis, and 84 had non-ASD developmental delays.
Hi, this is Summer, and I'll be covering the results found within the study that our group is sharing with you today. As stated before, there were 456 children involved in this research study. 238 of those children were previously diagnosed with autism, while 84 children had other developmental delays present. The remaining 134 children were considered control patients. The researchers did exclude from their study any children with unfilled cavities, dental diseases, or upper respiratory infections. The researchers analyzed small RNA molecules that are found in the saliva of 372 children with either a developmental delay or autism. Some of the RNA was also analyzed from the microbes present in each child's mouth. There were 5 types of RNA measured, which included microRNA and non-coding RNA. They used a machine learning algorithm which ranked molecules by their ability to distinguish the previously diagnosed autistic children from the 2 other subgroups of children. They ended up finding a unique set of 32 RNAs and further analyzed levels of these molecules in each saliva sample. Researchers evaluated the results of their test and found that it had correctly identified 41 out of 50 autistic children, 12 out of 13 children with developmental delay, and 18 out of 21 of the control patients. The probability within the results of the study showed that Clarifi had correctly identified autistic children with 82% accuracy and correctly identified children without the condition with 88% accuracy.
Conclusion. Our awareness of autism has developed over the last decades. In years past, this condition was just thought to be a developmental disorder with learning disabilities. Today, it is recognized to be a set of neurodevelopment disorders characterized by a deficit in social behaviors and nonverbal interactions. The families with ASD children have many challenges that they face. Unfortunately, some developmental obstacles are easier to recognize and diagnose than others. These parents need all the support and help they can get. With Clarifi, they can receive the help and support with just a simple swab of the mouth. The early diagnosis of autism treatment will be the most effective, giving parents hope for their children. Thank you again for taking the time to listen to our presentation. And does anyone have any questions?